So I'll be right with you. We still, for this unit, we still have to finish DNA replication. Then following that, we got protein synthesis. Then we've got, let me look at the drive, genetics. Engineering or not. I haven't yet this year with any of my grade 12s, but if we have time, we might. But if we don't, we won't. I got a feeling we probably won't. So I would say plus a gizmo on protein synthesis. So we've probably got 4 slides and a gizmo to do. So we're probably looking test-wise end of the first week of June, beginning of the second week of June, right? So around the 4th or the 7th, something like that. Who's missing? Okay, Rosa. Karema's not here. She's supposed to write. Callista, no. Teja, no. Patty, Miriam, Mandrew, Ramika. Okay, I'm gonna give, because we still have 3, 5, 6, 7, 8 people missing. I gotta give him a few more minutes. It's only 9:05. I'll wait till 9:10. So you can relax till that time. And in the meantime, I'll post the next bit. In terms of where we are, we're going to continue on with last 2 days ago's material. Just need to edit this in Classroom first. Topic, there is a topic. 28th, save. Actually, it may end up being next week. We'll have to see. All right, so present work, share. So we're on this DNA replication, still working on that. So if you want to go to that particular slide deck, and we are looking at we're on slide 16. It's this confusing thing, the thing that I have to do as a PowerPoint. Okay, so let me just open up that PowerPoint. By the way, the interviews last night were good. Okay. So here is where we are. Let me know when you can see the slide. Let's see. Am I presenting? Looks like it. Okay. that I've straightened, so it's not a helix anymore. Okay. The first thing that we have to do in order to duplicate this DNA is what? What do we have to do to the two strands? So step one of replication, which is what? Okay, and what's the enzyme that does the splitting? Don't worry about the single-stranded binding proteins and the gyrase. Yeah, helicase. So the enzyme is helicase. I'm not going to show it, but we're assuming helicase has come in and has split the two sides apart. So in that case, okay, so we've got helicase that split the two sides of the enzyme. Okay, now what that means then is we have to start. First thing we should do is label 3 and 5. Okay, which end is which? So can anyone tell me what the carbon numbers for these 4 ends are going to be? So is a phosphate a 3 end or a 5 end? So on this side, is the phosphate a 3 or a 5? Yeah, so this will be the 5. This will be the 3, 3, 5. Okay. So there's a 3 end there, 5 end down there, 5 end up there, and a 3 end up there, down there. So whenever you're working on a problem like this, after you split the DNA, first thing you should do is figure out which end is which, because it matters. Okay. Okay, which means in the new strands, you're going to have a 5 at the top and a 3 on the bottom on the right-hand side, because it's anti-parallel to the strand that's already there. Okay. Now, as I said, we need a starter or a primer already. I will explain later how that primer gets there. We're still not going to talk about that yet. Okay. So in the primer, I'm going to call that my primer. Okay. That's going to be my starter. It's something that DNA poly 3 can attach new nucleotides onto. Okay. So DNA poly 3 comes along. I'm not showing DNA poly 3 in this, but DNA poly 3 will be located right here. Okay. Keep going. Right. It explodes. And then they take off. Next one comes in. Binds. Okay. So this side is pretty straightforward. You're going to have continuous growth downwards. Okay. Because we're going from the 5 end of the new strand to the 3 end of the new strand. Okay. But what about the other side? Okay. If we look at the other side, 3 is up top, and 5 is at the bottom, because it has to be anti-parallel to the strand that's already there, okay? So if we try to do the exact same thing, if we put a primer at the beginning, at the top, this again, and bring in a new nucleotide, okay? When we tried to use the energy of this nucleotide to bond, watch what happens. The phosphate split off, the energy is released there, but where do we want the bond to form? Yeah, right here at the 3 end, right? So the energy for bonding is being released at the wrong position, right? So you don't get a link there. There's no energy to bond. So that doesn't work. Okay. So what's the solution? How are we going to get this to work? Yeah, we have to come at it from the other end. Yeah, you can. I wasn't sure if you're going to show up or not. Let me open up a break room for you. So start class, add a second breakout, open breakouts. You're at a desk you don't need. Okay. Go to group 1, that's the one we want. Okay, move this here. Karima, do you have the link for breakout room 1? Or do you need it? Just in case, I'll copy it. No, not the desk. There's actually a group 1 breakout room. So there's the link. And let me go to classwork and set up, reuse the post. Metabolic processes test, reuse it. Okay, it's going to Karima only. Due date is going to be today, which is the 28th. Time will be for 11:30 a.m. Make a copy. You okay? Assign. Okay, if you go to Google Classroom, and you go to this current week, you'll see this here. Let me re-edit that. Put your name on it. That's right. Okay. Save. Okay, so this is what you're looking for. Okay. Multiple choice questions are here. This is the written answer document. And Sorry about that. As Vedant said, we're going to have to start at this end, okay, down here, and work our way up. So there's our starter that's already there. DNA poly 3 is going to bring in a new nucleotide. We're going to have the system release energy and leave. Next one comes in. Energy is released. And they leave. Right? So one side, synthesis goes top down. The other side, it goes bottom up. But what do we do about this gap here? Yeah, we're going to have another strand or another section coming in from this side. So you'll have a start around there. Okay, you'll add another base. So the only problem we have is right there. This is because there's no bond at this point, and we need this to be continuous, right? So in order to link these two newly synthesized sections, we bring in an enzyme called ligase, okay? And ligase is an enzyme that will connect together any missing sugar phosphate bonds on the side of the DNA that's getting built in sections. Okay? Does that make sense? Do you understand how this goes? Is there any part of this you want me to go over again? Like I can restart the entire slide and start it up again. If you're unsure, if you want me to go over something, raise your hand. Okay. Also, in your textbook, if you're following along there, under molecular genetics, chapter 3, this is in chapter 6. We're on section 6.4, and we're roughly around page 285 is where we are. So 285, building complementary strands, is what we're talking about right now. Okay. Okay. I'm going to stop presenting this. I'm going to go back to the PowerPoint or the slide deck in Google Slides. Okay. So let me share that one. So what I just showed you in the PowerPoint is this mess here. Okay. If you have PowerPoint or a PowerPoint viewer, it's probably a good thing to grab. Okay, we're going to start here. Present. Okay, so we're going to talk about something called leading and lagging strands. Actually, you know what? This one isn't working very well either. I'll go back to the PowerPoint. Okay. Okay, so we're going to talk about something called leading and lagging strands. Okay, so here's half of a replication bubble, right? This is a replication fork. This section is yet to unwind, and I've got one strand up here and one strand down here. I am not showing you SSBPs. Okay, let's start labeling. So there's my helicase. There's my SSBPs. There's the growing replication fork. It's growing in this way. It's unzipping in this direction. There's 3. There's 5, which means the other end has to be a 5. into 3. Okay, let's start replicating. There's DNA poly 3. Okay, it's going to go on the lower strand. It's going from the 3. It's going to build a 5 here and add to the 3. Okay, and we said DNA poly 3 can only add new nucleotides to the 3' end. So that works. We're good. Okay, so the new strand will simply run this way, adding more and more nucleotides to the 3' end. The other strand is running 3'5. That doesn't work. DNA polymerase cannot add to the 5' end. It must add to the 3' end. Okay, so that being the case, DNA poly has to go in the opposite direction. Okay, so it'll start here and go in this direction. Then there will be another section that starts here and goes in this direction. And another one that goes in here and starts in this direction. Okay, so these little fragments of DNA that are made, okay, this is due to the limit of DNA poly 3. It can only build onto the 3 end of an existing DNA strand. It cannot add to the 5. So it has to build the other side in this manner. So the newly synthesized strands that are built in sections are called Okazaki fragments, named after the husband and wife team that basically discovered them. Okay. Okay, so that's Okazaki. The gaps between each Okazaki fragment will be where that covalent bond is missing between the sugar phosphate will be filled in by ligase. Okay, this side, which is built as one long continuous strand, okay, is called the leading strand. Okay. So the leading strand is continuous synthesis. The strand, the side that's made in sections is called the lagging strand. Okay. It contains Okazaki fragments, and the fragments are then joined together by ligase. Of the two, which one is going to be made faster? The leading strand or the lagging strand? Which one is going to be faster to build? Leading. This leading strand is called the leading strand. It's faster. So it actually goes along faster than the lagging strand. There are fewer steps. Okay? So leading strand will always be faster than lagging strand. Okay? So that's what this looks like. Okay, let's add on to this. What time is it? 9:30. Okay. No. Remember that this is half of a replication fork. There is more DNA on the other side that will form into the other side, right? Yeah, I'm only showing, this is the point of origin. I'm not showing you the other half. But it could be, right? It kind of depends. This also works. Let me go back. Yes, both sides do have the same problem. I will talk about that in a minute. Because from the point of origin, if this is lagging on this side, it's leading on this side. And if this is leading on this side, it's lagging on this side. The problem occurs both ways, right? Because it's unzipping this way. So Leading on this side means lagging on this side. Lagging on this side means leading on this side. We'll get there. Relative to the center of the bubble, yes. But remember, I still haven't got all the parts and all the enzymes in the system yet. We're still building this up. So I'm only looking at a replication fork right now. We're not even talking about the bubble yet. I'm just showing you what's happening in one half of a bubble. Okay? All right, give me my laser pointer back. Okay. So this is what's going on in terms of that question that Vedant just had. So there's my original strand of DNA. Okay. I've labeled my ends. There's my bubble. Right here, it's the point of origin, where my laser pointer is right now, where the red dot is. So one side goes that way, the other side is going in the other direction. DNA poly 3 on the right hand side of the bubble is continuous because it's going 5-3. So that's a leading strand. On the right hand side lower strand, it's going the wrong way. It has to go the opposite direction in bits. So that's lagging. Okay, so that side's fine. Leading strand. That side's lagging. But over there, it's leading. And up there, lagging. So from the point of origin, because of symmetry, as Vedant said, leading and lagging swap. Does that now make sense, Vedant? Okay. So everyone else, you have to understand, from the middle of the bubble, okay, leading and lagging will switch sides. Speed will essentially cancel out, yes. On one half, one side is faster than the other. I mean, in terms of what this really, I'll show you later, an animation. No. So there are 4 DNA polymerase 3s running on this particular model. And in fact, there'll be more than that, right? Because to be honest, there'll be one DNA poly here, there'll be another one here, there'll be a third one here, there'll be a fourth one here. So there should be, according to this lower picture, 1, 2, 3, 4, 5, 6 DNA polys, because there are 6 different strands of DNA. Okay? Okay. Is there any point in any of this that anyone needs cleared up to this point? Okay, can someone explain to me why we have a lagging strand? Why does one side always have to be built in pieces? Right, but why? Yeah, that's why, Vedant. Because this is growing, right? So this DNA poly wants to go this way, but as this opens up, all of a sudden we got a section that has nothing, right? Because when this part unwinds, this part with this strand up here will be exposed, but it's the wrong direction. So you have to add another DNA poly here and then start going this way to fill in the gap between here and here when it opens up. Okay? It's because of the way the thing unzips. Think of it like a zipper on a coat. As that so the zipper is helicase, and it's splitting up, right? So as it splits up, the lagging strand, the bit that this part that's currently closed, when it opens, all of a sudden you got to start from here and fill it back in. That's why lagging always ends up being done in bits. Okay, now Vadan had a really good question earlier, yesterday, 2 days ago, which was, well, wait a minute, how the hell does DNA poly then know how to start? If you, yeah, let me grab a jacket. Bigger. Stop sharing. And to make sure, I'm going to change my layout so I can see myself. Change my layout to spotlight. Sidebar, that works. Okay, now, here's my jacket. Okay, and there's my replication fork. Okay, my zipper represents helicase. Okay, now I'm going to assume this is the leading strand. Okay, so when I build the new strand, this is the 3 end. So the new strand is going to be 5. Okay, so on this end, as DNA unwinds, watch what happens. It unwinds, and this can continue going on this way. It's continuous synthesis. So that strand is fine. Okay, but on my lower strand, right, on this strand down here, it's going in this direction, right? So the point of the pencil is pointing in the direction of synthesis in the lagging strand. So watch what happens when I unzip. How am I going to fill in that gap? I can't go from a 5 end downwards. So I have to take a new poly and shove it in here and come from the replication fork this way. Then when that bit unwinds, I can't go this way. So I have to have a new poly come in here and start building in this direction. And that's why the lagging strand always has Okazaki fragments building up. Whereas this one, because it's going in the direction of this pencil lead, as it unzips, it just keeps trucking along. Okay? That's the idea. Okay, let's take the spotlight off of me. How do I unpin myself? Go back to grid view. Okay, is the laggy strand more? No, because the error correction that we'll be talking about later covers both sides. We'll actually talk about mutations as a separate PowerPoint. Okay, let's see. Classwork. DNA replication. Let's see if I can get this to show properly again. Okay, no, it's still not going to work properly. Okay, so I'm going to stick with my PowerPoint because that seems to be working better. Okay, so Vedant's question the other day was, how does DNA poly then know how to start these strands? How does it get going? Where do the primers actually come from? Well, it turns out, okay, remember, as I said before, DNA poly 3 can only build on the existing three end of an existing strand. So when you initially start, this is what the system looks like. There is no existing DNA strand. So we have to create something that DNA poly 3 can attach new nucleotides to. Okay? So we use primase. Okay? And what primase does is it adds primers. Okay? Or the starter nucleotides, about 5 of them. But what it adds actually is a little strip of RNA. So you get RNA primers that allow DNA poly 3 to have something to attach to. So primase knows how to initiate a strand. Poly 3 does not. Now that there's an RNA primer there, poly come along, attach its nucleotides to it, and just keep running. No, primase uses U. RNA replaces thymine with uracil. So in RNA, it's AUGC. All a U is, a uracil, is a methylated thymine, basically. So it's a thymine molecule with an extra CH3 group attached. So it'll substitute them in. Afterwards, it'll be removed, yeah, and replaced with DNA. And on the other side, you'll have a bunch of little primers that get put in by primase to give you the Okazaki fragments. So this is what I meant, Grant. I'm doing this slowly, bit by bit by bit, because it's complicated. So RNA primers are built by the enzyme primase, okay? And they serve as the starter sequence for DNA polymerase 3. You then have to replace the prime, because you can't leave RNA in the nucleus. So you have to remove the RNA and replace it with DNA. So ligase will come along. So ligase comes first, spot welds everything together. Then we'll use a different polymerase, DNA polymerase 1. And poly 1 will replace RNA bits with DNA. Okay. However, with eukaryotic, yes, that's right, Kieran. Eukaryotic chromosomes have another particular problem, though, that prokaryotic chromosomes don't. Okay. What shape do prokaryotic chromosomes have? What shape are they? Yeah, circular. Okay. What shape do eukaryotic chromosomes have? Oh, that's one pair of chromosomes. Yeah, they're straight, they're linear, they're not continuous. So if we come to the end of a eukaryotic chromosome, because there literally is an end because it's linear, see what happened here? The RNA primer was removed. Okay, so DNA poly 1 remove sections of RNA primer and replace it with DNA nucleotides, except at the very end of a linear chromosome, at the 5' end of a linear chromosome. It can't do that. Okay, because DNA poly 1 only builds, like any other polymerase, only builds onto the 3' end of a chromosome, of an existing strand. DNA poly 3 has no way of attaching nucleotides here because this is a 5' end. Why would that be a problem? Yeah, we're gonna. If this is a coding region of the DNA, if it makes a protein, you're screwed. Because all of a sudden you're missing genetic information that you need to do a protein. Protein won't work. Good guess about poly 2, Vidam, but no, not poly 2. And in fact, eukaryotes, in prokaryotes, there's only 5 polymerases. In eukaryotes, it looks like there's something like 15. Okay, so here's what basically happens. It's called chromosomal erosion. This happens in every eukaryotic cell. All DNA polymerases, it doesn't matter which one of the 15 you're talking about, or in prokaryotes, which one of the 5, can only add to the 3' end of an existing DNA strand. So every eukaryotic cell that you know of has this problem. So DNA poly 1 will come along, remove the RNA, and you're left with missing chromosomes. So this is what we call chromosome erosion. So as Fillmore says, Houston, we have a problem. You're also going to have the same issue at this end on the other side. Yeah, they are. Did I post them? If you look here, DNA replication, updated by the Amoeba Sisters, Crash Course, and Bozeman Science. So all 3 are located there. We'll take a look at probably either Amoeba Sisters or Crash Course after I've done all this. I also have a couple of other animations I possibly may be able to get through to as well. Stop. Window. Okay. So this means every time you replicate DNA, every time you go through mitosis, you're going to lose bases at the 5' end. Every single time. That means your chromosomes are getting shorter every single time your cells go through mitosis. Okay? Does this mean that there is a limit to the number of cell divisions that a cell can do? Because obviously at some point you're going to start eating too much information in the chromosome that it will no longer work. Okay? Is there a solution to this? There actually is. So we have something called telomeres. Okay? Telomeres are repeating non-coding sequences at the end of a chromosome. So it's this black region here. Okay? It's like a protective cap. So The dark blue area is where your nuclear genetic information actually is. Then you get a whole bunch of random nucleotides stuck here. And their job is simply to get shorter every mitosis round, every replication, and not get into the information that's actually needed. Okay? It limits the number of cell divisions for any nucleus to be about 50 cell divisions. We kind of do a little bit. Okay, so this is the protection. This is how they fixed the end. We were using a special enzyme. I'll get to the enzyme in a minute. Okay, the enzyme that, yeah, basically, every round it'll be a little bit more unpaired. Okay, so the enzyme that builds the telomere is this one called telomerase. Okay, and it is the enzyme that extends telomeres. Okay, this is the only enzyme we have that can add bases at the 5' end. Okay, however, its activity in mature cells basically is non-existent. Okay, so in your muscle cells, in cells that are specialized and differentiated, muscle cells, stomach cells, skin cells, hair cells, brain cells, nerve cells, uh, cardiac cells, it doesn't matter. Telomerase activity is almost zero. Okay, so it does not stay active throughout your entire life. It's a short-term solution. Where you have highest activity of telomerase, stem cells and cancer cells. Okay, why would it be really high? Why would you have lots and lots of telomerase activity in stem cells? Because they need to divide so rapidly. And cancer cells, one of the reasons they're cancerous is because they reactivate telomerase, which keeps extending the telomere and therefore ends up protecting the cancer cell from losing any genetic information, which suggests a potential way to attack cancer cells. If we could figure out how to turn off telomerases in cancer cells, then cancer cells would end up eating into their genetic information and the cancer cell should probably die. We haven't figured that out yet because we can't be that specific. If we use drugs that affect telomerase in a cancer cell, they also affect telomerase in skin cells, hair cells, and stomach cells, which is why people who are on cancer drugs often feel nauseous, lose their hair, and take such a long time to repair wounds. No idea. Evolution's completely random, so who knows, right? Evolution isn't goal oriented. Evolution just evolves until it's just good enough to function, right? It's not survival of the fittest. It's survival of the just good enough. If it works, great. Go with it, right? Evolution works completely differently from the way we humans design things. I mean, when we build things, that's basically it. That's why we have, you know, such crazy evolutionary systems in our bodies. I mean, the entire human female reproductive system is nuts. There's no way a good engineer would ever design the way human females give birth and reproduce, or at least give birth. That's a silly system. it's broken. It's actually probably an evolutionary dead end. Other systems that don't work or are not designed very well, um, systems like the way we breathe is vastly inefficient, right? Because we use a concurrent system, not a countercurrent system. Yeah, to me, they have the longest neuron of all land animals. It's about 2 meters. Well, we all do that. That's an evolutionary history. It's the vagus nerve, spinal nerve, cranial nerve 10. So in evolutionary history, cranial nerve 10 came out of the base of the skull and would go down into one of the 6 gill arches. So the where the gills are in fish. Okay. And what happened with that vagus nerve is that vagus nerve got co-opted into other functions. So it initially starts out going down towards where it originally was supposed to go, then realize, oh wait, I'm in a giraffe or a human, not in a fish. So I'm supposed to turn around and go back. So I have to actually go back up the neck. And so yeah, you're right. That vagus nerve 10 in a giraffe does go down that long and then all the way back up. Yes. Because it's initially following the original evolutionary pathway, then realizing, oh wait, no, I'm supposed to be doing something else. It could be. But remember that, well, I think you're stating it wrong. It's variation and not evolution, because adaptation is evolution. Yeah, absolutely. The absolute was to cure it, not the Vedant. There are huge ethical issues with that. Yeah, I don't think anyone can ever know. Okay, it's 10 o'clock. I know your head's probably swimming a bit. Why don't you take a 10-minute break? 10:02 actually. So let's say we get back together at about 10:12, 10:15. Say 10:15. Okay, so I'll see you then. With this. All right. Before I do that, I'm gonna play a video that I think I will add actually to Classroom. So let me add this. Add YouTube. Control C. URL. Control V. Add. Save. Okay, so I'm going to show you a video now. That talks about DNA replication. Go full screen on this.
The replication of DNA begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I, which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The current view of DNA polymerase III is that the two subunits function together with the DNA on the lagging strand, folding to allow the dimeric DNA polymerase molecule to replicate both strands of the parental DNA duplex simultaneously. Proteins other than DNA polymerase III are not shown.
Okay. Um, do you want me to show you guys either the Amoeba Sisters or the crash course in real time? It's fast. In fact, I can. No, not that one.
The replication of
Let's see. Long. Okay, let me find. Where is it? This is from
Using
Harvard.
computer animation based on molecular research, we are able to see how DNA is actually copied in living cells. You are looking at an assembly line of amazing miniature biochemical machines that are pulling apart
Leading
the DNA
strand,
double helix and
lagging
cranking out a
strand.
copy of each strand. The DNA to be copied enters the production line from bottom left. The whirling blue molecular machine is called helicase. It spins the DNA as fast as a jet engine as it unwinds the double helix into two strands. One strand is copied continuously and can be seen spooling off to the right. Things are not so simple for the other strand because it must be copied backwards. It is drawn out repeatedly in loops and copied one section at a time. The end result is two new DNA molecules.
That's about that's real speed, right? I mean, remember in one of your cells that are going through mitosis, it's about an hour to completely do all of mitosis. It's fast. It's probably about 20 minutes to copy your entire DNA in preparation for mitosis. I mean, as the narration lady basically said, that helicase is spinning as fast as a jet engine to unwind the DNA. I remember that in each cell, You got 2 meters of the stuff. So it's fast. Okay. I think Amoeba Sisters is usually pretty good. So let's do Amoeba Sisters and then we'll finish off this slideshow, I think. Sorry, you have to deal with this ad for a sec.
Video creators. No matter what video you're creating, Envato Elements has everything you need.
DNA. We talk about it so much. It's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where. Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when. When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, that's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. Now remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has 2 strands, you could think of helicase unzipping the 2 strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes...
So when she was talking about topoisomerase, that's the enzyme I called gyrase.
RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has 2 strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3', and the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strand. Now, the thing about DNA polymerase is when it's building a new strand, it can only build the new strand in the 5' on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase, once again, is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have two identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the two copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors. In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA bases, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving medical treatments that can stop DNA replication in harmful cells, including pathogenic bacteria or human cancer cells. We encourage you to explore beyond the basics. Check out the further reading suggestions in the video details to explore more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
So again, that's again a simplification because it didn't go into the different kinds of polymerase, but it gives you a good overview of what we've been talking about. And none of the videos that I've ever found ever touch on telomerases. Okay, just so you're aware. All right, so let's look at this diagram. Okay, so we're looking at a replication fork. Okay, and the replication fork is unwinding in the direction of the pink arrow. Okay. So that green blob is helicase. The little round light blue circles are single-stranded binding proteins. Okay. In pink are the primase enzyme 3. On the lagging strand, in both pink and blue, are the Okazaki fragments, where pink parts are the RNA primers and blue is the DNA part of the Okazaki fragment. The light blue L over here is ligase. That's basically putting together the fragments. DNA poly 1 is coming in, replacing the RNA with DNA. This side is synthesized continuously, so is the leading strand. This side is synthesized in part, so is the lagging strand. Okay? So be sure you know the functions of all of these different enzymes and how this, in terms of how this works. 3 does this. Y is one side leading, Y is one side lagging. Okay? So take a look at the videos, review these notes. This link down here no longer works because no website supports Adobe Flash anymore. So ignore that link. It doesn't work anymore. Okay. What's sad? That the Flash links don't work? So DNA polymerases. Okay. DNA poly 3. Okay. Works at a rate of about 1,000 bases a second. Okay. So it is the main DNA building enzyme in the cell. This is kind of related to Abby's questions about how fast the system works. DNA poly 1 is the one that replaces RNA. It also proofreads. So it's a spell checking enzyme. That's what the Amoeba sisters meant when they said DNA poly also acts as a proofreader. Well, they were talking about a different version of polymerase, poly 1, 20 bases a second. Okay, so it's involved in editing, repair, and primer removal. So it's the sun discovered poly roughly around this a few years later. Okay, has it cleared up? Or am I still frozen? Okay. So poly 1 was discovered by Arthur Kornberg. Poly 3 was by Thomas, although I don't have the exact year that Kornberg did it. Okay. Close the door. Okay. So regarding editing and proofreading DNA. So let's say that there is something wrong. Like let's say that 2 thymines that are next to each other end up binding to each other instead of binding to the adenines across. Okay. This is what we call a thymine dimer. Okay. So if you're going at a rate of 1,000 bases a second, the probability of having spelling errors or typos is pretty high. Okay, so poly 1 comes along. Okay, and proofreads and corrects the typo, removes the errors. Okay, so it'll repair mismatch bases and it'll remove abnormal bases like the thymine dimer. Okay, and it does this throughout your entire life. Okay, so it reduces the error rate of poly 3 from 1 error in every 10,000 bases to 1 error in every 10 million bases. Right, and so this is just showing you, you don't have to know this diagram, it's just an example of how poly 1 would repair something. Okay, but we're reducing the error rate by quite a bit, from basically 0.0001 to 0.6 zeros in a 1. Okay, the system is fast. Okay, E. coli bacteria will copy 5 million bases in less than an hour. Okay, and then divide to form 2 daughter cells. Okay, human cells will copy the 6 billion bases and divide it into daughter cells in a few hours. Right, we do it fairly quickly. It's incredibly accurate. Okay, we get a mistake every 100 million bases. Okay, so you're thinking, what does that actually mean? How many errors are we actually getting then? It translates to about 30 errors every cell division total, which is not much. And yet, with 30 errors every cell division, and you guys have gone through a lot of cell division, how many mutations do you think you carry? How often do you get cancers in your lifetime? Right? Or how often do you mutate? Yeah. Right? I don't think anyone in here is an X-Men. Okay? And that's why, if you think back to Mendel, we are diploid organisms. Even with an error rate of 30 errors per cycle, even if you get a mutation, usually mutations are going to be recessive. They're not dominant. And since you have 2 copies of every gene in your body, if one goes off, you have the other one that can basically cover for it. It's the advantage of being a diploid organism. In fact, if you think about it, 50% of your genes are probably heterozygous. You've got one copy that's probably not working properly in half your genes in your body. Okay? And that's just from inheritance, even before we talk about cell division. So that's pretty good, right? We have a pretty decent redundant system. It's not perfect. If it was perfect, we wouldn't have cancers and other mutations. Everyone would basically be, pretty identical. So why do we still need errors then? Why not make a perfectly replicating system? Yeah, we need that variation to evolve, right? That's why we have to have some errors creep in. Otherwise, evolution would have nothing to work on. So this is what it would actually look like, right? Here's one replication bubble. Here's a second one, a third one, and a fourth. You can see one linear end of a chromosome here, and there's the other linear end down here, right? So this is a eukaryotic chromosome because it's not circular. It's not a loop. Okay, that's it. We'll finish 10 minutes early. I would strongly suggest you use your asynchronous time to look at some of the videos and review this stuff, because this stuff is not easy. And I've still got a whole bunch of stuff to add to this. So take this weekend to really review this, because I'm adding more complexity to this next week. Because we go from here to protein synthesis, and then we talk about how we control both systems. Okay? All right, so you are dismissed. Have your good selves a good weekend. Please review this. Take care, all.